Hi everyone. Today I'm going to start a 7-day mental challenge on reading one scientific article a day. So this is a 7-day read a scientific article challenge. Here I have mentioned it just as an article because maybe you're not interested in science and you're interested in any other field. You can read any article of your choice, but you have to do this consistently for 7 days. In my challenge, I'm going to take the 7-day read a scientific article challenge. Now I'm going to talk to you about what I hope to get by the end of the 7 days. At the end of the 7 days, I'm going to hope that I have more information on the topic of my interest. I expect that after a week, it is going to actually help to increase my memory function. It would help me to process more information in relatively small time from future on, and I want to making the reading of scientific article fun for myself, and that is why I'm going to take up this challenge. And today is the day 1 of my workout. This paper that I'm going to read is on a protein called ATVCCN1. I'm going to get into the information pretty soon. I have included the name of the paper, the author's name, and their affiliations. I'm going to put the link of the paper below in the description box for you guys to take a look at the paper whenever you wish to do so. Thylakoids are present in plant cell chloroplast, and these cells adjust to light intensity for photosynthesis. They do so by influencing the proton motive force, also known as PMF, and this PMF is present across the thylakoid membrane. It is believed that chloride channels are involved in maintaining the proton motive force, also referred to as PMF. One such chloride ion channel is VCCN1. Since this VCCN1 is going to be discussed in the paper, this VCCN1 that I'm going to be referring to here will also be referred as ATVCCN1. I refer to this as ATVCCN1 because this was derived from a plant called Arabidopsis thaliana, from which it gets the initials AT. The importance of this protein is in the fact that it can protect the plants from high intensity of light and thus acts as a photoprotective agent for the plant. Photosynthesis machinery is present in the thylakoid membrane, and the PMF is present across the thylakoid membrane. So in the thylakoid membrane, you have a stroma region and a lumen region. The thylakoid membrane forms a barrier between the stroma and the lumen region, and the electron transport machinery that is present in the thylakoid membrane. So this membrane is dependent upon the concentration of ions that are present from stroma or in the lumen region. The transport of ions results in the proton motive force. Proton motive force has two parts, a delta pH part and a delta psi part. Let me get to it in a moment. Charge separation occurs due to light intensity, and hydrogen ion transfers into the lumen of the thylakoid, and this generates the proton motive force in the thylakoid membrane. The proton motive force is composed of the change in pH. This will be referred to as delta pH, and that indicates the hydrogen concentration gradient. And the change in the electric potential gradient is also referred to as delta psi. and this delta psi is dependent upon the charge. Both of these components are important for PMF and drive the ATP synthesis in the chloroplast using the protein called ATP synthesis present in the thylakoid membrane. ATP is the energy equivalent required to run the metabolic functions in the organisms. When the H+ concentration in the thylakoid lumen increases, the pH decreases. This reduces the electron transport through the cytochrome B6 complex component in the photosynthesis machinery. This means now the PMF is less, so there is more energy for electron transfer, and this energy is dissipated out as heat by the process called non-photochemical quenching, which is a protective mechanism for plants to save from high intensity light. Depending on the light intensity, the components of PMF, the delta pH and the delta psi, can vary, and this is known as the PMF partitioning mechanism that actually helps in fine-tuning the photosynthesis. So the PMF can be regulated by either the amount of delta pH or by the amount of delta psi present at any moment. Generally, plant stores PMF as pH to downregulate the electron transport and activate the NPQ at high light intensity. However, in low light intensity, this PMF is reduced to store as pH to maximize photosynthesis and downregulate the NPQ. The million-dollar question is, why should we study ATVCCN? To begin that, let me remind you, PMF has two components. One is the delta pH, the other one is the delta psi. Both delta pH and delta psi depend upon the ions present and the kind of ions they are, the charge of the ions, and how much they are present. There has been study on other ionic channels such as potassium channels. But the study about chloride ion channels has not been evaluated as much before this paper was published. And with that respect, the authors bring to you that how chloride ion channels can actually affect the photosynthesis mechanism and how they affect the partitioning of the proton motive force or the PMF. How they regulate the PMF that finally goes and regulates the whole photosynthesis mechanism. And that's why we want to study about chloride ion fluxes that happen. Now, where does this name ATVCCN come from? The AT comes from Arabidopsis thaliana. BCCN stands for voltage dependent chloride ion channel. In this paper, they have discussed about ATVCCN1 and ATVCCN2. For my presentation purposes, I am mainly going to focus on ATVCCN1, which is the most talked about protein in this paper. I highly encourage you to go through this paper at your leisure to figure out the different experiments they have done and the results. However, I am going to discuss about the results of this paper in a few minutes. This ATVCCN1 protein is important in fine-tuning the proton motive force and adjusts the photosynthesis with respect to variable lights in plants, and that's why it is important to study the ATVCCN1. Chloroplast is the main organelle for photosynthesis. Do we know that if ATVCCNs are located in chloroplast? In order to know that, the authors performed an experiment. What they did in that experiment was they fused the ATVCCNs, in this case both ATVCCN1 and ATVCCN2, with green fluorescent protein, also known as GFP, and expressed them in knockout mutant strains of VCCNs, which were referred to as VCCN1 or VCCN2 mutants. And in these mutants, they overexpressed this GFP attached VCCN1 or VCCN2 gene under the CAMV35S promoter. If you look at the figure A, you can see the green fluorescent protein. And from chlorophyll fluorescence, you can visualize the chlorophyll present in chloroplast. When the GFP attached with VCCN is expressed in the chloroplast, in that case, after the merger, you should get the yellow color formed by the combination of the green and the red from the GFP part of the VCCN and due to the presence of the chlorophyll that was already fluorescing. In the control, since there was no VCCN expressed, you do not see a merged image that shows the yellow coloration. Whereas for the VCCN1 GFP and the VCCN2 GFP, you do see the yellow colored ATVCCN1 and ATVCCN2 proteins being expressed in the chloroplasts that look like yellow in color over here. We also want to know that which part of the chloroplast does this VCCN1 or VCCN2 go and localize. In order to do so, they have performed immunoblotting with help of antibodies that are raised against the GFP. Remember that the GFP is attached to the VCCN1 or VCCN2. So both GFP and VCCN1 or VCCN2 are being expressed. And on immunoblotting with the anti-GFP antibody, they should be able to locate the presence of VCCN1 or VCCN2. And as you can see in the figure B, you do get the whole protein with the GFP bound in the chloroplast and in the thylakoid. But then which part of the thylakoid? So if you look at figure B, the CLP stands for the chloroplast, the THL stands for thylakoid, ENV stands for the envelope, STR for the stroma, and STR.L for the stroma lamella. Each of these different fractions or the subfractions from chloroplast have a specific kind of protein that can act as markers for them. And the purity of those fractions can be known from the specific protein that is already in that fraction. Now, if we raise an antibody to that specific protein that is a marker for that subfraction, we should be able to know where the GFP is localized. And based on the immunoblot that you see in the images B and C, it is evident that these ATVCCNs are present in the stroma exposed region of the thylakoid membrane. Next, we want to look at the electrophysiological importance of this protein ATVCCN1. And for that reason, the authors put the recombinant ATVCCN1 in planar lipid bilayers and then saw the results that were generated from these proteins in the lipid bilayers. The following image shows the electric signal that was produced on transfer of chloride ions. If you look here, this protein is selective for transferring chloride over potassium. In these images, the O's stand for open channels, whereas the C stands for closed channels. A permeability ratio between chloride over potassium showed there was a 1:0.17 ratio between chloride to potassium, which means more of chloride ions are permeated through these membranes. This shows the selectivity of the channels for anions such as chloride over cations such as potassium. This channel can also help to transfer nitric oxide at very low levels, and that has been showed in the supplementary of this article, but I do not discuss about it over here. These results also show that there is a fast gating and a flickering pattern that was observed at positive voltages that you can see in the upper panel, whereas the burst-like openings were dominant at negative voltages, as you can see in the lower panels. So we also see that there is a cooperative gating that is present, which means at the same time more than one channel can open, and this openings can be as many channels. So there can be opening of 1, 2 or 3 channels. So now we know that ATVCCN can help in transfer of chloride ions through the membrane by the help of this ionic channel. But if this anionic channel is inhibited by a blocker such as DIDS, in that case we see that the electrogram does not show the signal that was present due to the chloride channels. This channel is not affected by cationic inhibitors such as tetraethylammonium, which suggests that ATVCCN1 is definitely a anion channel. In presence of light, there are hydrogen ion accumulation in the thylakoid lumen. This causes the delta psi or the electropotential gradient to increase in the lumen side, and that causes chloride ions to influx to the thylakoid lumen using the protein such as ATVCCN1. As the chloride ion influx in, the chloride ion's negative charge can neutralize the positively charged ion of hydrogen, and this leads in a decrease in the electropotential gradient of delta psi. So as the hydrogen ion concentration decreases in the lumen, the delta pH on the other hand increases inside the thylakoid lumen, and that can affect the ATP formation by the ATP synthase enzyme present in the thylakoid membrane. So now we know that light can actually affect the delta psi, right? But the point is that how is VCCN modulating the PMF and how it is helping the acclimation of the photosynthetic machinery from dark to light. So the change in the PMF size and how much of delta psi is affected over the delta pH is affected is dependent upon the amount of chloride ions that is influxed in by the ATVCCN, and that can actually affect the ATP synthase, the activation of NPQ, and the other events involved in photosynthetic machinery. In order to understand how the ATVCCN1 chloride anion channel is affecting the PMF size, an experiment was done using electrochromic shift. In this experiment, during the elimination of intact leaves, dark intervals was provided in order to see how the chloride channel ions actually affect the PMF size. For this experiment, 3 specific varieties of plants were taken. One was just the wild type as it is. The second one was a VCCN mutant. And the third one, called OEVCCN1, stands for overexpression of the GFP tagged VCCN1 protein plants. The VCCN mutant plant will be referred to as VCCN1-1. As you can see from figure A, the PMF reaches a maximum value within the first 2 minutes and then decreases by 20% till the next 5 minutes and then remains stable over the rest of the time period. If we compare the wild type with respect to VCCN1-1, which is the mutant VCCN, we see that the mutant VCCN has a elevated PMF size as compared to the wild type. And in case of the overexpressed VCCN1, this PMF size is actually decreased compared to the wild type. This decrease that we see for the overexpressed VCCN1 could be due to accumulation of the negative charge of the chloride ions in the lumens. And this presence of the chloride ions in the lumen may eventually help in preventing the decay of the ECS signal when the lights are turned off during the dark phases of this experiment, which is required for the estimation of the PMF. And that's why the graph for the overexpressed VCCN1 looks so different from the wild type VCCN. Now, if we look at the figure B, we see that there is a high amount of delta psi, or the electropotential gradient, maintained at the expense of the delta pH in the VCCN1 mutant, which is VCCN1-1. When we say a high delta psi, this is always with respect to that of the wild type. And if we look at the overexpressed VCCN1, in that case, we see that the whole PMF that is present is entirely due to the pH and not due to the delta psi component of the PMF, which means that in overexpressed VCCN1, the pH component of PMF is getting completely uncoupled from the delta psi component of the PMF, and this indicates the delta psi uncoupler behavior for the overexpressed protein. This actually kind of suggests that on overexpression of this protein, the delta psi is dissipated when light falls on the plants. On illumination, this delta psi is dissipated, and that's why there is a significant delta pH that is present across the thylakoid membrane, as you can see clearly from this figure number B. Using the electrochromic shift assays, when we start the experiment, after that, in order to figure out the hydrogen efflux rates present for these 3 different plant varieties, the experiment was done in order to understand how much of hydrogen ion is effluxed out of the membrane. It was seen that in the mutant, there is a higher rate of efflux. Remember, in the VCCN mutant, we do not have VCCN, which means there are no presence of chloride ion transfer through the specific VCCN ion channel. And in case of wild type, with respect to the overexpressed VCCN1, we see that initially the H plus efflux rate is reduced in the overexpressed VCCN1, but later it increases over that of wild type despite the total reduced PMF size. Next, the chlorophyll fluorescence was studied in order to evaluate the NPQ. NPQ also stands for non-photochemical quenching, and NPQ decreased in the VCCN1 mutant and increased in the overexpressed VCCN1, which resulted in an adjustment of the linear electron transport measured as the photosystem to quantum yield, as you can see in the figure E at the bottom of the image. So now this makes sense because now ATVCCN1 is involved in the light triggered voltage dependent chloride fluxes in thylakoid, and this contributes to the counterbalancing of the hydrogen pumping that is happening across the thylakoid membrane. What happens during the photosynthetic acclimation from low to high light? When there is a transition from dark to light, in that case the VCCN1-1 mutant that does not have VCCN displayed a lower, slower NPQ induction, but without any significant changes in the linear electron transport compared with the wild type. So remember that for looking into the linear electron transport, we are looking at the photosynthetic yield in the panel B, whereas panel A shows us the results from the NPQ. In the overexpressed VCCN1, the NPQ is induced faster and the decay is slower, resulting in less of electron transport than we see for the wild type. This pattern of change that we see both in the VCCN1 mutant known as VCCN1-1 and OE VCCN1 can be explained by the observation that ATVCCN1 activity helps in building up the delta pH, because remember there is more of chloride ion channel opening for chloride ions to pass to the lumen, which actually increases the delta pH and suggests its involvement in fine-tuning the photosynthesis on transitions from low to high light. The aforesaid results could be depicted in a model system. So when there is initially light, that can be represented as the grayscale in this model. During the initial response to the light, there is an increased delta psi. However, this also leads to the chloride channel opening. So chloride ions keep on coming in, and then the delta psi keeps on decreasing and an increase in delta pH occurs. And that affects both the NPQ and the photosynthetic yield of the plants. Previously, ion fluxes have been hypothesized to modulate changes in the thylakoid membrane architecture. So in order to look if there is any thylakoid ultrastructure changes due to presence of ATVCCN1, there was studies done using the wild type Arabidopsis thaliana leaves using transmission electron microscopy versus studying the Arabidopsis thaliana leaves for the mutant VCCN plants, also referred to as VCCN1-1. In case of the mutant plants, there was very long grana stacks in the dark system. And in the light adapted system as well, the granas were long and also they were banana shaped as compared to the wild type. This suggests that the thylakoid membrane network is rearranged in presence and absence of channels such as VCCN. With this, we reach to the conclusions of this paper. From this paper, what we learned today was that ATVCCN1 is a voltage-dependent chloride ion channel. It can help transport of chloride ion from the stroma region to the lumen region of the thylakoid through the membrane. And this ATVCCN1 protein is homologous in a lot of different organisms, suggesting that it can actually act as a chloride flux. This ATVCCN1 regulates the proton motive force by influencing the delta psi component as well as the delta pH component of the PMF. The delta psi component of the PMF is the voltage that activates and regulates ATVCCN1 to transport chloride into the lumen, which then results in partial dissipation of delta psi in the thylakoid membranes and increases the delta pH. ATVCCN1 can reduce the size of the total PMF, and in that way it can regulate photosynthesis. Overexpression of ATVCCN1 can lead to an increased rate of hydrogen efflux through the ATP synthase, despite a lower PMF than in wild type, and without impacting plant growth under standard condition. So this is a good point for the development of ATVCCN1 in plant systems. And even a lower PMF size could be sufficient for the activity of ATP synthase during steady-state photosynthesis and plant growth. ATVCCN1 overexpression results in PMF consisting solely of delta pH and enhanced acidification of thylakoid lumen, and supports higher NPQ and lower electron transport. So we can use ATVCCN1 as a target for improving photosynthetic acclimation in presence of light and dark for agricultural and bioenergy production. After reading this paper, I really liked this paper, and I might not have done a justified job in explaining this paper, so I definitely urge you to go and read this paper. This is a wonderful paper. These people have done a lot of work on this chloride ion channel, and it gives us a lot of insight into how chloride ion channels can be involved in the photosynthetic machinery by influencing the proton motive force that is present in the thylakoid membrane, which forms an integral part of the chloroplast that is involved in photosynthesis. I really liked this paper, and this was the day 1 for my 7 days challenge of reading one scientific paper. Here I have created a chart for myself. If you want, you can even download this chart. And once you have made a presentation of a paper after reading it on a specific day in a week, go ahead and put a tick mark against that day. If you want, you can take a printout of this chart for influencing yourself in order to make yourself read a paper every day. I know sometimes it's pretty hard due to the hectic lifestyle we have, but it is possible to read a scientific article or any article of your choice in a day. So in order to make this a good habit and just to get yourself into a 7-day challenge mode where you read articles that you prefer or that you really need to read for a specific occasion, you can go through this process. I hope it helps you in reading a paper every day, And as for my first day, I have ticked into the first day of my reading a paper and presenting it. Have a great day ahead. If you like this video, please make sure to put a thumbs up and subscribe to my channel for more videos. Have a good day ahead. Bye.